because this is the first structural biology lab plus we are having.
No.
C++ labs we are having this semester.
What?
C++ lab course and Java lab, these two labs we are having in this semester exam.
Okay. What about structural biology? Have you registered the paper?
Yes, sir. Structural biology lab we have registered.
The structural biology, yeah, almost all of you have registered. So this semester itself, you will write the exam for this paper, right? Okay. So anyway, in that case, actually, some of your class, the practical class, will deal with what? Wet lab work. Okay. So regarding like UV spectrophotometer, okay, CD spectrophotometer, and crystallization and purification and DNA and melting point, all these things that you have to carry out in like wet lab. Like only few things are we can do in online. Okay, so the torsion angle calculation, all those things that you have to do in that lab only. Okay, but the thing is like we don't have like a virtual lab, right? So we are conducting like online lab only. What I'm saying is, whatever that we have to do in wet lab, I will do it as the concept explanation. Okay. So what is mean the UV, what you are going to do in the UV experiment. So those things like I will do explain conceptually. Okay. So based on that, I will conduct the test. All right. Then like whatever that you have to do in online. Okay. So those things also I will give you like a demo, and so within this that we have to stick and we have to conduct the exam. All right. So I'm going to take I'm going to explain the experiments, what are the concepts behind that, okay, how you are going to do all those things. So maybe like for each experiment, okay, I will try to give you the notes for each experiment. Okay, for some experiments, like I will give you the notes. For some other experiments, based on my class, whatever that I'm taking, you have to prepare the notes, okay, and you have to submit it. Okay, once I am telling you like completely compile all those things that you have to compile, and for experiments that you are doing like online now, so those experiments that you with result, whatever that you are doing with result, you have to combine and compile it and together you send to me. All right. So today we are going to see about like this protein folding problem. Okay. So the first experiment is like denaturation experiment. I don't know how many of you have syllabus with you right now. I do not know. So learning the protein folding problem, the first experiment. So in that you are going to denature and renature the protein, and then you will be doing like a UV study of denatured protein. Okay. So from that, what we are going to do. So that's what we are going to see today. Okay. So in order to understand this, we have to get an idea about like protein denaturation. Okay. So what is mean by protein denaturation, or like what is mean by folding problem? Okay. So you all know that central dogma. So once the DNA is coded, okay, so the DNA coding will be like translated into like a protein sequence, right? Then the DNA synthesis that will bring like each and every amino acids in the ribosome, right? Then all these like one by one, the amino acids will be coined, and finally you will have like a polypeptide synthesized. Okay. So if you have like a protein of like 500 amino acid, so 500 amino acids will be coined, and it will be a polypeptide chain which is having 500 amino acid, a long polypeptide. Okay. So this polypeptide, before it is getting into like a native conformation, right? Maybe like a globular protein or like some other extended form, whatever. The native conformation, before it is attaining the native conformation, it is a linear polypeptide chain, right? So something is instructing for the polypeptide to coil, okay, in a like particular orientation and to have like a particular shape. Okay. So this process is called like the folding. Okay. So folding into the native state. Okay. How this polypeptide is folding? So that is the folding problem. So far, like so many theoretical explanation is there, but till now it is not like a completely understood. Okay, maybe like if you go into this Brandon and Tools, okay, folding problem, they might have explained you very in detailed manner. Okay, whatever that I am discussing is like just part of that, because we don't need all those things. If you are curious to learn more about, you can study that book and you can learn more in this subject. All right. So how come it is like folding it? Right. So who is telling that this is the native conformation? Right. So each and every second, like thousands of proteins are right synthesized. Thousands of polypeptides are like being synthesized continuously, and each thousands of polypeptides is folding into its native conformation. How it is possible? So it's a very complex process. So far we do not have like any idea, but theoretically we have like some idea, and that's what we are going to see. Okay. So the process by which a polypeptide chain acquires its correct 3D structure, and it should become biologically active. That is called the native state. Okay. So not only like a folding, okay, it should be biologically active. Otherwise, there is no meaning, right? So biologically active. That once it is folded into native conformation, the protein should be ready to do the function. Okay. So how it is doing? So that is the problem. It's a protein folding problem. So you can assume that this is like a long polypeptide chain. Okay, which is like unfolded. All right. So from unfolded, once it's going in this direction, right, and it becomes like a native conformation. It becomes like a folded protein. Okay. So you can think of like once the polypeptide is synthesized is moving into the direction of folding. Once it is getting like a native position, it's not denaturing. It is not coming back to the unfolded state. It's not coming into the linear polypeptide. Right? So when it is like a polypeptide, it is very fast to fold into to attain the native conformation, but The reverse reaction is not true. The reverse reaction is not happening, right? So we are seeing in our body, thousands of proteins are like attaining like a native conformation. It's not going back. This is like very stable thing. It is unstable. The unstable condition, right? Once it is attained like a native conformation, the time taken for the native conformation is very short, very well in a nanosecond level. But it is not coming back to the unstable state, unfolded, or like a linear polypeptide. So once it is attaining like a native conformation, what are the factors to hold this polypeptide into the folded conformation? Again, like thousands of questions unanswered. Right? So all these things together, it's called like a folding problem. Okay. So you should understand that in order to understand this folding problem, you should understand the structural features of the protein. So we have discussed all these things in the last our theory classes. In detail, I have told you the protein is having like a hydrophobic core, and on the surface you have like a hydrophilic surface. Okay, so the protein is making like a hydrophobic, preserving the hydrophobic core inside, right? All the hydrophobic amino acid, all the hydrophobic side chains will be facing towards the center of the protein. Hydrophilic side chains, hydrophilic amino acids will be exposed to the solvent. Right? So even some hydrophilic amino acids are present in the hydrophobic core. The charge will be neutralized by having hydrogen bonding with some opposite charge. Right? So all these things are like a structural features which is preventing the protein. Okay, which are the factors preventing the protein going back to the linear polypeptide form. Okay, so these structural features are like very important for making a linear polypeptide into a globular protein. Okay, so all these things that I have explained in like previous classes. So here you can see this is like a unfolded protein. Okay, so you have like a linear polypeptide chain, and so the linear polypeptide chain, it will be in a random movement, right? So always it's not going to be in this shape. So it will be bending here, here, it will go here. You know the Brownian motion, right? So it will be like a very dynamic motion, the unfolded state. Okay, from here it's going to the globular state. So it's called like an intermediate. It's called like a molten global state. It's somewhat resting position. Okay. So compared to this unfolded state, the dynamic, okay, the movement somewhat will be rest. And from once it is attaining like a molten global state or like an intermediate state, it's ready to fold. Okay. It's ready to become like a properly folded three-dimensional shape. Okay. In a molten global state, if you see the structure, okay, it will be attaining almost all the essential secondary structure. Wherever you have like alpha helices, so it will form a alpha helix. So you can see these are like a beta sheet, beta strand. Okay. So the alpha helical secondary structure, the beta strand, everything is like almost it is made. The only thing is the beta strands are not coupled. It is not making a beta sheet, right? So it is in a properly folded state. You can see these two making like a beta sheet, these two making like a beta sheet, and it will be like a hydrogen bonded. So here you have like a strong hydrogen bonding. Here you have like a strong hydrogen bonding. Everywhere you have like strong hydrogen bonding, which is preventing the native form of the polypeptide. Alright. So there are like theoretically several proposals. how it is like, say, you are getting like folded state. So you can see here like several unfolded state. Okay, thousands of confirmation of like unfolded state. Okay, and the unfolded state, the number of unfolded states is getting reduced when it is in a molten globular state. Okay, it's in a molten globular condition. The conformation, the number of conformation is somewhat reduced. When it is finally achieving like a native form, is getting like a only one folded state. Okay. So here you can see the free energy map, where you have like a very high energy for the unfolded state, and molten state, molten globular state, you have like a little bit like a lower in energy. Okay, when it is reaching the transition state to attain the folded state. Okay, so it little bit the high energy is like high, but at the folded state, the energy is like very less, and you have like a very stable conformation. Whereas these are like unstable condition. Okay, so once it is crossing the energy barrier, the transition state is reaching the folded state, which is like a very low in energy. Okay, so there are like different ways of folding. People like suggested different way of like folding. So some people suggesting like a single step. Okay, the protein is folding into a single step and it is attaining the native state. Okay, some others suggest like two different way, not like a single way. Multiple ways are like being adopted to attain the native conformation. Okay, so whatever it is, but essentially you are having like a intermediate. It's called like a molten global state. Okay. And then you can see like in some of the proteins you will have like a disulfide. Okay. So how this folding is correctly recognizing the disulfide bridge, right? You have like a cysteine residues, right? So this cysteine, maybe it can go and make a disulfide bridge here, right? This cysteine may be going into this state to make like a disulfide bridge, but it is not doing like that. It's correctly choosing its pair, and it is helping to attain the like native conformation. How is it achieved? How it is achieved? Okay, still like a lot of like unanswered questions. Okay, so here like again like several proposals. You can see like this is like a polypeptide which is having like 55 ml. This is like bovine trypsin inhibitor. So you have like 1 to 6 cysteine residues. So there are like 3 disulfide bridges in the native conformation. Okay, so 5, 14, 30, 38, 51, 55. Here you can see 5 and 55. Okay, 30 and 51, 14 and 38. So this is the native conformation. Why it is not making like 5 with 38 or 5 with 14 or 5 with this 30? Why it is not making? How it is selecting 55 its partner? Okay, so it is like very complicated question. Okay, so here you can see like first it is making like a 30 and 51. Okay, so once it is making 30 and 51, it can go either this way or this way or this way. All right, so all these confirmation you are getting like some percentage amount. Okay, you are getting like 10%, 20%, whatever, right? But only the native confirmation. Okay, only whatever it is the right pairs, the disulfide pairs, that only will be a highly stable, which is helping to attain the original native conformation. Okay, so here like protein disulfide isomerase, it is an enzyme, okay, which helps to make like the disulfide exchange, which enhances the rate of reaction. Okay. So apart from this, okay, this folding like thing, apart from that, you have like a molecular chaperones. So these chaperones, you can see this is like GOES, GOEN chaperoning, okay, which are missing to make a protein into a like a proper folding. Okay. So this GROEL and GROES complex you can see here, which is a protein machinery. So unfolded protein that will go into this chamber. When it is coming out, it will attain like a proper folding. So simply like the cartoon explanation you can see here. So this is like a protein machine. Okay. So you have like unfolded polypeptide chains entering. Okay. So this ATP, this energy is given by using the ATP. Okay. So once it is going, you can see like a conformational change in the protein machine. Okay. So once it's like a conformational, it's doing like finally you will see like the unfolded protein will come out as a native conformation. Okay. So chaperoning is also helping to attain the native conformation. Okay. So this is like a general idea about like a protein folding process. Okay. So now we will talk something about like protein denaturation. Right? So just hold on a second. I will come back in 5 minutes. Okay, just stay on online. I'll come back. Students, you were able to see the screen, right?
Yes, sir.
Yes, sir. Yes, sir.
Okay. So we are going to see about like what is mean by protein denaturation. All right. So how you can do the denaturation? Can anybody say? Do you have like any idea that whether you will be able to denature the protein?
Altering the pH.
Altering the pH. Okay.
By heating.
By heating.
or by using some detergent.
Detergents, okay.
And, as Satish mentioned, by adding some increasing or decreasing pH, so we can denature.
Okay, so that's good. So by changing temperature, pH, or you can add like a urea, something like that. Okay, so that will denature the protein. Okay, so what I'm doing is I am giving you a protein solution. Okay, so I am giving you a protein solution, and I am asking you to find out whether the protein is in native form or in a denatured form. So is it possible to find out whether the protein is like a native form or in a denatured form? Can anybody say?
Sir, we can run onto the gel, base gel. If we have we know the molecular weight and our band is around that particular band size, so we can say it is in native form.
Yeah, you will run the gel, but you will get a form, you will get a band. But how
Yes,
do you know whether it is in a native or in a denatured form?
Sir, whether it's checking the activity of the protein.
So you need to, you are telling activity.
So in case of case, if we are using sucrose to break down, then it is in native form. If it is not, it is in denatured form.
Yeah.
And we can use Western blotting for to check whether this protein is expressing or not.
Western blotting.
Yes, sir. Just to check whether this protein is expressing or not. If it is expressing, that means our protein is in native form.
No. The thing is like we have a very limited thing. Okay. Don't go for like higher end experiment. There are like so many experiments are there. Okay. Like simply, can I find out by using like simple instruments?
Yes, sir. Using the spec UV.
Very good. Yeah, you can use like UV. Hold on a second. Yeah, tell me. So by somebody told that by using the UV spectrophotometer, we can find out. So who told that?
Sir, it's me, Subhash.
Okay, Subhas. Okay, so tell me, Subhas, how you can learn by using like a UV spectrophotometer?
Sir, the optical, activity is like around 280 nanometers for proteins. So it is like usually the observance of the aromatic amino acid present in proteins.
Okay.
This absorptivity is based on the aromatic amino acids present in the protein. So that range is fixed at 280 nanometer. We will check using the quartz cuvette. Protein solution is added to the quartz cuvette, and then and the UV at 280 is measured.
Alright, so But how do you tell whether it is like a native or in like unfolded state?
Sir, a spectrometer gives only protein concentration. So it do not differentiate whether it is in native or not, in unfolded state. We
That's
can't
what I'm
cannot
asking.
measure.
That's what I'm asking. We'll get
So
absorbance.
yes, sir.
Based on absorbance, how will you tell?
Sir, based on the absorbance, we can't say whether it is in unfolded state or in native state.
Okay. Anyway, so you guys told somewhat like partly correct. So we will discuss that. All right. So I'll tell you now. So you can see here, okay, the effect of temperature on enzyme action. Okay. So at your the rate of like enzyme action, okay, is increasing with the increment of the temperature. Okay. So the rate of enzyme action will be like keep on like increasing, but at particular point it suddenly fall and it begin to denature. Okay. So after that there won't be any enzyme reaction. All right. So each protein is tolerable for like particular amount of temperature, beyond which it's not stable. So at a particular point, the protein will become like denature and it will lose its native conformation and it's not going to function properly. Right? So, so denaturation is a phenomenon which involves transformation of a well-defined folded structure of a protein to an unfolded state under non-physiological conditions. Okay, so it occurs suddenly and completely over a narrow range of conditions. Okay, but it can be reversed. So initially, like in the beginning of the class, I told you like a polypeptide, it will attain like a native conformation, right? So the reverse, like once it is like a denature, it can go back to the original native form. Right? So what are the factors? I told you like several hydrophobic factors. Hydrophobic interactions are there to hold the protein into like a native form. Okay, so only one native form is there, whereas you have like a thousands of denatured unfolded state is there. If in the unfolded or like a denatured form, you do not have like a proper disulfide bridge, you do not have like a hydrogen bond, you do not have like any hydrophobic interactions, all right? So nothing will be there. Right? So hydrogen bonds, electrostatic interaction, all those things, okay, are the key factors to hold the native structure. Okay. So here you can see like several proteins having like a different range of thermal stability, right? So even some proteins are there which can survive at a very high temperature, right? So here you can see like a concentration of like a denaturant or like a temperature. The temperature is increasing here, or the concentration of denaturant. Urea is keep on increasing. The percentage of native structure, native folder. Okay. Low concentration, low temperature, 100% native conformation. Up to a critical point. Okay, up to a critical point, the protein is stable, after which it suddenly collapses and it is becoming like a polypeptide. Okay, so temperature, organic solvents like pH, so everything, pressure, so all these things like influence the denature of the protein. Okay, so thousands of like polypeptide conformation. So if you see the energy, okay, so only like one native conformation which is having like a very minimum energy and it's stable. All right. So whereas in a polypeptide you will have like multiple secondary structures, but like unstable. You have a very high energy. Right? So the conversion of a biological function molecule into a non-functional form. So there are like many denature states, but only one native state. So protein can regenerate to their native state, but slowly. So denatured proteins have like a greater tendency to aggregate. Okay. So far I was talking about you like the denaturation process. So now we come back to our question. Okay. So Subhash told you can utilize the UV spectrophotometer in order to find out like a protein, whether it is in a native form or in a denatured condition. All right. So this I will tell you. So suppose I am adding like urea. Here, take a protein. I am adding urea. So the protein will denature. All right. And what I am doing is after denaturation, I am doing dialysis. Now removing urea. Okay. So then it is coming back to the native conformation. Okay. So it will come back to the original three-dimensional structure. Okay. Now again, there is no chaperone, there is no other assistant. All right. You are removing urea. That's all. But once it is removed, it's coming back to the native fold. So now who is telling the polypeptide to go back to the original native form? So all these things comes under folding problem. All right. Okay. So you are having your protein. What I am doing is I am adding like 10 millimolar urea, and then in another tube I am adding 100 millimolar urea. In another tube I am adding 200 millimolar urea. Okay, and in another tube 500 millimolar, and in finally maybe like 1 molar urea or 2 molar urea, whatever. All right. Students, you are following, right?
Yes, sir.
Okay.
Yes, sir.
So now I am adding urea at the increasing concentration for each tube. The protein is same concentration. So what will happen? The protein will start to denature. Okay. So as a result, what will happen? So I am adding like a 10 millimolar urea, which is like a very low concentration, and then 20 millimolar, and then 50 millimolar, 100 millimolar, 200 millimolar. So for each tube, if I am going to check the UV spectrophotometer, so what will I get?
Sir,
I will get the absorbance for each tube differently. Yes or no? For
Yes,
each
sir.
tube, the absorbance, whatever that I am getting, will be not same, not equal. I will be having difference in absorbance. Okay, it is because the reaction timing is dependent upon the concentration of the urea. Okay, so I have the native protein in which I have not added any urea. Okay, so I will measure the absorbance. Say the value is X. Okay, now I am measuring the absorbance in the tube. Like I have added 1 molar urea. 1 molar urea, huge concentration. So that immediately it will denature the protein. Okay, so the absorbance will not be equal to X. Am I correct?
Yes, sir.
Yes. So the absorbance will not be equal to X, for which is the concentration of the native protein, the original protein. Okay. So I am checking for a 1 molar concentration and then 500 millimolar concentration. So for each cube, if I see the absorbance, definitely you will have variation. Okay. If you see the 10 millimolar urea, it will be somewhat closer to the value of X. It's because the urea concentration is very less, where you have the native population. The protein in native population will be somewhat closer to the original protein. So you will have the absorbance close to your native protein. But if you see the absorbance of the 1 molar urea, right? What will you see? The native population will be very less. So definitely there will be some difference in absorbance. Yes or no?
Yes, sir.
Yes.
Yes, sir.
So now I am telling you another fact. So Subhas told we can measure the UV absorbance. It is because of the aromatic amino acids. So that's true. Okay. So the aromatic amino acid, okay, in a native form, it will be inside the protein. It will be in the core of the protein. Okay. So I mean the aromatic amino acids will not be exposed. All right. In a native form, the aromatic amino acids will reside inside the core of the protein. They are not much exposed. Whereas in a denatured form, the aromatic amino acids will be exposed. Okay. So will you see the difference in the absorbance or not?
Yes, sir. We can find
Definitely you will
Yes.
see the difference. Okay. So by simply, okay, you can observe the difference between native and denatured protein by using the UV spectrophotometer. Okay, and you can run column chromatography. Okay, in order to identify like protein is like a native form or in a denatured form, you can run column chromatography. Okay, so in a column chromatography, you will get like a different retention time. So based on that also you can see. But as far as like our experiment, we are using the UV spectrophotometer in order to find the native or like a denatured condition. Okay, so this is the idea behind the usage of like UV spectrophotometer. In the UV spectrophotometer, okay, so Subhas told like around like 280 nanometer, you can measure the absorbance. So I'm putting like another question. Okay, in case in my protein, if I don't have aromatic amino acid, Okay, so the phenylalanine, tyrosine, tryptophan. So these are like three amino acids, which are like aromatic amino acids, which will absorb around like 280 nanometer. So all the proteins does not have these three amino acids. Okay, there are some proteins which do not have these aromatic amino acids. Okay, so in that case, if you want to measure the absorbance at 280 nanometer, is not going to help you. So what will you do?
Sir, maybe we should do wavelength scan with the protein sample we have, and then we can fix the higher peak one.
So in that case, again, like we have to depend on like UV spec only. Okay, since you don't have like aromatic amino acid, it doesn't mean that you cannot measure. Okay, so you have the peptide bond. Okay, NH, right? NH single bond, C double bond O. You have the peptide unit. So this peptide unit will absorb at around like 190 nanometer. Okay, so if you have some absorbance around like 190 nanometer, so definitely you can say you have the protein. Okay, just 1 second. So you have like near UV region, for UV region, and you have like a vacuum UV region. Okay, so you will have the electron transition from sigma to sigma star, n to sigma star. N means like a non-bonding orbital. Okay, and then pi to pi star transition, and n to pi star transition. So these are like four different transition level that are possible. Okay, so in our case, we have like a possible transition of like pi to pi star and n to pi star. So if you see peptide bond, you have the double bond where the pi electrons can have the transition. Okay, so even if you don't have like these amino acids, aromatic amino acids, it's possible to measure the UV spec for polypeptide. All right. So I have given you like a rough idea about like a protein denaturation and renaturation and the usage of like UV spectrophotometer. Okay. For unfortunately, we do not have the lab session. So if we have the lab session, maybe we can have like a experiment conducted. Okay. So whatever that I have told you. Okay. So I will give you as a send you a file of notes. So you can keep this as a record. Okay. So within this, you may get a question for your exam. All right. So maybe I can stop here, and for the next experiment, and we will see in the next week. All right.
Okay, sir.
Okay.
So if you have like any questions, you can ask me. Otherwise, we will sign off.
Sir, do we have to write in a copy notebook?
Yes, you need to write a record.
Okay.
I will send you notes for some of the experiment. So based on the notes, you prepare a record. Okay. So some of the experiments I will not give you the notes. So for that, you have to prepare on by yourself. Okay. Combine everything together and then you send it to me. All right.
Okay, sir.
So, okay, guys. So we will see next week. All right. See you then. Bye.
Thank you, sir.
Thank you.